Good morning. I hope you can hear me. Is it fine? Then my duty is to start this course with some kind of introduction, and I will concentrate my introduction on the structural aspect of RNA structure. And all the structural aspects, although I know that structure is clearly related to function, so function is depending on the structure. So I will start from the very basic background information. So I know usually the audience here is rather dispersed in terms of background. So we don't be afraid if things are too simple for you. They may become more complicated this afternoon or tomorrow. So it's good to start from the beginning. And the beginning, some background. These audiences very well aware of these. All the information, the basic principle of molecular biology and information related to the content of DNA goes from DNA to protein. So it's a unidirectional information content. And the nucleic acids are very key players. And when we speak about function of nucleic acid, it's always related to translation, transcription, and transfer of genetic information. So nucleic acid and genetic information are really the key principles. And you will see it goes DNA. You see the arrow here means that you can adapt. You can play with DNA. And RNA used to be concentrated at the beginning in the translation process, going from RNA to protein. But you will see that RNA is a much more important player than just for that part, although that part is key. In terms of genetic information, what do we have? Here we have, you see the human genome, complete sequence, and divided in subgroups in terms of information content. What kind of information do we have? And you see that the protein coding part is very small. It's less than 2%, which means that 98%, almost the global information content of the genome is related to other aspects than just the information content of the gene sequence. And so most of it is regulation. And then there are other activities. Some of them, I mean, a lot of these functions are not yet known. But in terms of just the gene contents, you see that the coding part here, I hope you can see actually, exons part is very small compared to the non-coding segment. And why is that? Because it is very important. The control, the regulation part is the essential part in life. Without regulation, the information content will be totally useless. So then let's go a little bit further and come back to RNA in a more realistic way. What is the contribution of RNA in terms of molecular biology of the cell this time? So before, we had this sequence of events going from here, DNA to protein, that way. Very simple. But then you see that RNA, now you see really the implications of RNA in the whole process of cell biology. You see that RNA is involved in transcription through regulation, through epigenetics, several things. So that's why you see arrows going both ways. So RNA is implicated. RNA is, of course, implicated in translation. That's a classical view. You will hear several talks about that, how going through RNA from sequence information about the sequence to from the genome to the protein, really. And here, so these multifunctional aspect of RNA become really crucial, and you will see that here we reach one key point, is that RNA has a regulatory function. For example, you have non-coding RNA, which are functional RNA molecules, which do not code for a protein. Most of them actually are in that category. RNAi interfering gene expression, which were discovered in the late 90s, and the microRNA, that you will hear also a lot about, were discovered about 10 years ago. So now we will go a bit further in terms of structural aspects as this background, and you will see that there are two types of interaction which characterize RNA structure. One is the RNA function controlled by Watson-Crick base pairs. And it's the most classical, the base-base pairing. And the other one where the function is controlled not only by the base pairing, but by the architecture. And when you see the architecture, you have several aspects that we'll go through very quickly, very briefly. So first, here you have a short glimpse on a few structures that give different aspects of the diversity of RNA in terms of structure. Here you have the structure, the genome structure, classical type of helix, the double helix, which is the most simple. We'll come back on that helix, because even helix can be quite flexible, quite variable, and the detailed structure is very important in terms, especially that the helix is part also of a more complex structures. In fact, the architecture of RNA is an assembly of small pieces of helices. And then the way they assemble, they form an architecture. For example, one of the first one will come back a bit more on that, because it's a key molecule, and not only key in terms of functional importance, but also very interesting because it was the key to unravel the architecture of RNA. It was the first RNA structure, functional RNA structure, that was determined and that could define the characteristic rules. And everything we know about RNA structure has its origin in the tRNA structure. And here you have a bit more complex structures. You see at the bottom here the ribozyme. That is a small RNA, but with an enzymatic activity. That was also very new. So RNA can perform catalysis without proteins. That was a very important discovery. And you see this is a very small molecule. When you compare the scale is about the same here when you compare the two. And you see that's another view, because when we have the tendency to present schematic drawing, when you have the complete CPK type model, the full model, you'll see these two are the same. The one at the bottom here, the ribozyme, and the left is the same molecule, just the way to see it. As you will see, it's much better to try to follow the schematic drawing than the full picture, which is very difficult to follow. But we'll come back to that. And we're talking about, we're still in the overall presentation about RNA structure and function. So the architecture of RNA is important, as you will see, but the different structures are important because that gives hints to the recognition problem. What is functionally important in a structure is what the structure can perform. I mean, the molecule can perform in a given structure. We saw the ribozyme. And then also the peculiarity about each structure gives it a specificity of recognition. And that's a very important functional step. So here we have some examples of complexes. So functional assemblies are complexes, and they are either nucleic acid, or most often, in terms of function, nucleic acid protein complexes. And here you have just a few examples of complexes here where you see a helical stem here being recognized with the protein on the left, which goes through the main groove. Here, just an idea of how these different structures with the same or almost the same helical stem view, these complexes are different just because here you have a recognition which is from a major groove here and here on the shallow groove. So just a different side of the helix and you have a different recognition stem. We'll come back to that. And here you see when you have helices, like in this D form, the last, I hope you can see with my dot, the right left, bottom right here, you see helical proteins that recognize the RNA helices. And you have a recognition of grooves and backbone here. And here you have even more complex structures on the left here with the recognition and assemblies. Sorry? The pointer. Yeah, the pointer has a real problem here. Okay. I will skip because I don't want, I have a time problem. I think I will concentrate more on that. That was the introduction. So now I still go on with some chemistry on the structure. And first, one important, we're going to see a few functional aspects linked to the structure. So first, nucleic acids are negatively charged biopolymers. That's a very obvious thing. And so the charge is on the phosphate part. The phosphate groups carry the negative charge. So that's the DNA type of sequence, ATGC. And for RNA, of course, you have a U instead of the T. And, but it's exactly what is important is there are two key informations in that slide. First, the negative part charge are on the phosphate. So when you see the backbone, very often the negative part is on the backbone, and then that creates a charge attraction, negative attraction for protein. That's why you have positively charged group that interact. And then the other aspect, very important to keep in mind, yeah, that was, is the polarity. This is one aspect which is very often overlooked. People, nucleic acids have a polarity. It is very important when you decode. We are not going to concentrate now on that, but you will see when you're talking about decoding aspect, regulation, and specific sequences, the polarity is a key specificity of nucleic acid structure. And you find that polarity and asymmetry are key structural specificity of biological molecules that you have always to keep in mind. These molecules are, when you look in that direction and in the other direction, you see different molecules. You see different aspects. So the sequence, as it is there, and the reverse sequence, 3', 5', if you reverse the sequence, you will see you have a different structure and different recognition. And life is always playing with this aspect. That's really the specificity of biological molecules. Where do we have, that's also just a small jump in terms of information content. Where do we really have found all the information I'm talking to you about? You know, I'm showing molecules. I will show you even more later on structures. You will see today, tomorrow, complex structures. Most of that information, exclusively, almost all information available is experimental information. We are not talking about models only. Actually, they are very, very seldomly models only. we are talking about experimental information. And here you have an example of that experimental information, which is the result of crystallographic work. So crystallography was key in bringing information about structure of proteins, of nucleic acids. And here you have a clear information, and you have a glimpse on the improvement of the quality of the information during the years. Here you have the same base pair. For purpose, I wrote to you a non-classical base pair, a GU base pair. So that was the electron density, the grid here, available in 1985. Long time ago, but not so long after. 3 angstrom resolution. That's the resolution when you see things. And 1999, 1 angstrom resolution. 1 angstrom is already very high. Now we can go for proteins and biological molecules up to 0.6. 0.6 is really the limit, because it's roughly half the wavelength. That's the theoretical limit that you use for X-ray to investigate. So 1 angstrom gives you the grid. So the information content is the electron density that you see here in the grid. You see when you had that grid, the model was very important in terms of interpretation. For example, here, the density you could move a little bit. You had some flexibility. So the theoretical interpretation and the modeling, the computing aspects of that was key in terms of refinement. At this level, there is no interpretation. You see exactly the position here of the interacting atoms. You see the dots here represent water molecules, because crystals are hydrated. And then you have the picture. And so you can see that in the GU base pair, and we'll come back later, you have this wobbling. And here are the 2 base pairs. I'm using that because you will see, at least tomorrow, the day after, I'll give you a presentation on the ribosome aspect of the decoding, on the latest results, as you'll see. I mean, some of them are in press, so it's not only historical, so you will have previews almost. And this GU aspect in the decoding is key. It's the wobble. And you will see that having the structural information at the experimental level forces to reinterpret our thinking and our theoretical interpretation about decoding. So what I'm going to tell you now is based on this type of data. Of course, very simple. And tomorrow, Wednesday, you will see the latest application of the GU base pairs in the decoding. I mean, the importance of the GU base pair as a key. Okay? So don't sleep on that. Don't forget. So let's come back to more basic things. So these are the 3 groups, chemical groups, that forms the basic unit of nucleic acid, the phosphate, the ring, the pentose ring, and the purine of pyrimidine. The phosphodiester bond is what links, I already mentioned. On DNA, you have ATG here. On RNA, you have the U. And you have also the other key important factor is the hydroxyl group, which is here on the 2'. So the U is important, but the important thing is this hydroxyl group. The stereochemistry now, always keep in mind we go 5', 3'. Nucleotides from I to I+1. You have this organization of the phosphodiester, which gives you all the links. And all the angles here are very important. We will see how. The conformation of the sugar base. The this is anti. You see that when you look compared to the ring here, pentose ring, this is the position of the base, yes, where in the syn, the purine is shown, but the syn conformation can exist for all type of one, is the position of the sugar with respect to the base is rotated, you will see, the other way. So this is the most common structure, but in some cases, not in helices, of course, but in special three-dimensional specific architecture, you can find syn organization. Sorry? The syn, the rotation of the base, the syn is rare. Yeah, but you can find in some architecture, three-dimensional complex architecture. The normal one is the anti. Now, one glimpse on the sugar pucker, what is called the sugar pucker. What is a sugar pucker? Pucker is you have a pentose ring, and when you have the ring, if you build a pentose, you will see you can flip. It has some dynamics. Here you have example. For example, this is the ring. This carbon group, the C2 prime, can be up. If you take these 4 atoms, can be up or down. And according to that, it's just when you take the ring, you do that. You will see it flips like that. It's a very flexible conformation. It's called the sugar pucker. It's the flexibility of the sugar which allows nucleic acid to adapt to different contexts. So it's a very important movement. And in terms of consequence, look here, what is presented there is just playing with only the sugar pucker. You will see when you play with the sugar pucker, this is the dynamic that you see. So you can adopt a wide distribution of these positions of the bases and the phosphate compare, just by doing that. So that shows how dynamic are these structures. And by the way, if I say that this pucker is very important, you can imagine now the importance of changing one hydrogen group on this ring by an hydroxyl group that is going from DNA to RNA. So with the DNA which has no one hydroxyl group less, then the flexibility is more important than for RNA. Because the OH group of RNA induce some rigidity and also some anchoring possibilities. So here is the conformation, the standard conformation RNA helices, which is the C3 prime endo. You see how that's the C3 prime endo. It's like that. The C3 prime group is up compared to the base of the ring, and the anti-conformation of the base. The C2 prime endo, which is a non-standard conformation, and is not found in helical conformation, only in complex three-dimensional structures. It's non-helical region. You see that if you move that, the C2 prime, instead of being up, is down. And you see that the distance here between one phosphate and the next one here goes from 6 angstroms to 7 angstroms. So just playing with the pucker here, you extend, sorry, I was finding my word, you extend the distance between the phosphate group. That's why you don't find that in regular helices, because if you would play with different distances, you will distort helices. So that conformation gives the A form, I remind you, C3 prime endo, A form helix, C2 prime endo, B form helix. Okay? But you cannot pass from one to the other. Another interesting feature for structural feature that you can find in the with the nucleic acid is the Watson-Crick pairs are isosteric. What does that mean? It means that the distance between the anchoring point of the base to the sugar is the same, regardless of the base pairing. That's very important to form an helix. Can you imagine if you wouldn't have that, you would have bulky things, it wouldn't work. It's an interesting feature. That's why you see here a regular helix is formed. That's a consequence of this isosteric. And by the way, I showed you before the GU. You can keep that with the GU by wobbling. But the distance can form. So you can form. But there are small distortions. Now another important aspect of the helices which results from the base pairing, classical, is that one side of these pairs, that is this side where the phosphates go, is the major groove or the deep groove, and the other one, which is facing the ribose part, is the minor shallow groove. So one is here and the other one is here. A more simple view, nice. And that is a very important. aDNA or RNA is the same principle of architecture, except that you cannot have bRNA, sorry. You cannot form, you could, would be very costly energy-wise, so it doesn't exist just because of this pucker I told you before. Okay? So RNA, what is characteristic of this aDNA structure, which is a consequence of the Parker and Halsey, is that you see the major groove here as a very narrow entry. It's very close. Compared to the bDNA, where the major groove is the most accessible part. That's a very important difference. You know, in a regular helix, you cannot reach the major groove bottom. That's why in terms of interaction, the recognition, and functional aspect of RNA, you don't have very long stretches of helices, continuous, in terms of function, because these are, the major groove is not accessible. If you want to access the major groove, because we will see later that the major groove is where you have the most interesting recognition sites in terms of bases, the chemically accessible part. So if you want that, you have to open, so you have to distort. Or you have to be at the end. You see at the end of the helix here, if you finish the helix in just a connection, then this change becomes different. So that's why the end part of the helical stems are very important in terms of recognition, are a very important functional aspect of RNA. That's not true for DNA. DNA, you don't have to unleash it until the last steps of transcription, of course. But you can recognize the DNA in the form, in the helical form, in the bDNA, because you can access the major groove and you can read the base sequences. But here you cannot. Okay, that's a very important part. In terms of 3D vision, is like that. Here you have the volume exclusion. You see that the major groove here is very accessible, and here, practically nothing. Now, where are the bases? Here you have the sequence. This is all GC, the model. And you see also another aspect of between RNA helix or aDNA. I said that vRNA does not exist. The same is true. aDNA is not very common. You have, in very specific cases, conformations of DNA which are A, but not long stretches. And you see the difference here. For example, in terms, the distance is almost comparable. But in one case, if you would look through here, you see only one base. They just stacked on our. Where here, the bases are along a groove which you can see through. Round. What I said before, in terms of accessibility, you see here it's easily accessible. Here it's not accessible. So if you want to access these and to make a complex, here you have a complex type. The complex can come here very easily and distort. So the complex part here is something in between, because you have inserted a molecule. That can be any molecule or a fragment of peptide or something. And you form interactions. So either you shrink here if you have, or in order to access, you don't enter here. In order to enter, you have to have some distortion of the RNA. So that can be achieved through another complex. So you can bind another part which distort the RNA structure, and then you access it. That's one way. That gives you a sequence of events. But practically speaking, if you want to enter that, it cannot. It has to change. Here it can, and then it changes. So the sequence of event in terms of function is really structure dependent. So when you know the different structures of complexes, that gives you snapshots, and then you can interpret the sequence, the real sequence of event, the kinetic and the dynamic of the functional aspects through these snapshots. I just trying to fold. So now the RNA folding, that becomes more important. I'm talking about, up to now I spoke about very basic structural aspect, that is base-base and helices, and you see already the specificity of RNA. What is very unique with RNA is the folding. So you have the folding, which means the RNA can form 3D molecules, which are made of piece of helices, but connected, and the three-dimensional structure is not just an helix. While with DNA you have helices. Helices can go around like in nucleosomes, and then you can build that. You can open, but you don't form complex, stable complex molecules with DNA. DNA is the carrier of the information, but the RNA is really the functional worker. So a good example of three-dimensional folding, as I told you, is given by transfer RNA, which is a small molecule, but with a three-dimensional structure. So here you have the 2D sequence. You see stems. So you have 4 stems, 4 helices, and these can be described as I described before. They are RNA type or aDNA type structures, and these can be. But what is now very unique and exceptional is that these stems are assembled to form a three-dimensional structure. This famous L shape. And you see that you can find, if you follow these loops which are connecting, you see the loops here positions the stems the way they are. And in fact, you have 2 stems, one here and one here, unique to form the L shape, with here one accessible loop which carries the information. We'll see tomorrow and the day after the importance of that. That's the part of the message, the anticodon. That's the decoding part. And here is the part which carries the message. And these are important, functionally important, but because they stabilize this L shape. So when with this molecule you have a three-dimensional aspect, you see on one side, as it is not very wide, it's like a helix. The size here is just a helix almost. So you see how functionally that is important, because when you come to the ribosome, you have to align them. And if you would have bulky things, you can imagine the ribosome would be totally different. And the structure would not be unique, then it would be very difficult to carry on the translation process, you see. So the three-dimensional important, just this basic principle shows you how each piece of structure here is key. Because you want that, and then you want the specificity. That is, each tRNA molecule has its own specificity. Each RNA specific molecule, ribosome or other, it's not only just a graph structure, it's not just an architecture. it brings the right information at the right position. So that's the importance of all the structure and function relationships. And this distance is key. This conformation is key. Remember what I told you about the accessibility RNA structure? If it would be just a stem, you would not access this part. But it's the end, then you can access it. That's where you have the information content here. And this is to stabilize. And here. So here you come back with the color code which shows you the 4 stems, the anticodon, the CCA terminal that gives you the 2 loops. And just a glimpse, we'll follow later. The tRNA is so important that you will see all over the traditional common function. So I show you one picture that you may not see too much later, is a complex with EFTU. You see here the tRNA, that's another view. That's the groove, that's the shallow, that's a major groove. You see, it would not be very much accessible because here we have we don't have all the space feeding model. The groove here is even more shallow. And here is the EFTU. EFTU binds here. Here. That's a specific position. I don't enter in that, but you know, binding here leaves it's very far away from the anticodon. It's not, of course, it's close to the CCA end, but it leaves the tRNA almost free to interact with everything else. So it grabs it. And the fact that it binds here is not just, like that. You have always to keep in mind that this is just a snapshot part in a movie which has several plays. And when TFTU binds, it binds on the tRNA which is bound to the synthetase. So it has to access a part which is accessible. And it's just that trivial question shows that you don't have tRNA floating around and then, oh, I bind it. It doesn't work like that. You have one complex, it moves on one side, and then it has to be recognized. It has to be to have some specificity, but not unique specificity, because all tRNAs have to be recognized by EFTU in the same way. You see the complexity? So you need specificity, the 3D specificity, but not too much specificity, because then you would be in trouble in terms of function, because EFTU is carry all tRNA, not just one. You see how you play always with specificity. So the 3D structure gives you some specificity. The sequence information, that is the base content, can give you more or even more specificity. At this part here, the sequence is essential. We'll see that later. In this part is important, but cannot be too specific. We have a display with structure and function. Now, just a few glimpses. We move a little bit on another structure, more recent one, a riboswitch, where you have another 3D structure where you find always the same principle, the stacking. It looks like an helix, but it's not. I mean, it forms an helical type of interaction, but you see the chain here is very different, interacting. So, but you tend always to form the same type of association and to minimize things. And that gives you either more rigidity or more flexibility, depending what you want to play. And here is the sequence there. And of course, this is larger and more flexible, accessible. Now, I will finish very quickly, because I try to convey the message that there is more than just base-base recognition. So you will see in the application presentations, especially on the ribosome tomorrow, how you can make use of this information in terms of function. And now just to give you a more general overlook, which is an ontology for base pairing, which has been designed by Eric Westhoff, and then he you can classify the type of interaction, the nucleic acid, in order to grab the information on first Watson-Crick pair from standard RNA and define the 3D structure, non-Watson-Crick pair from 3D RNA module responsible for RNA recognition and 3D fold. And of course, all these are important for RNA recognition by proteins. So the interacting edge are here, here, and here. So it just shows you when you have a base, you have 3 accessible parts. That's very simple. And if you are on the Watson-Crick edge, it's here. And we come back to what I told you before. This is the Hookstein edge. Hookstein edge is not normally accessible, but there are Hookstein bases, base-base interaction. When you say it's a Hookstein base, it means that the interaction with the other involves this part of the base. And you can easily imagine that you cannot form Hookstein bases with just a canonical structure. You have to change. So for the Hookstein base, you have another type of interaction. And the sugar edge is, tells you how, which part of the interaction you are dealing with. For the, yes, this was the purine. Now for the pyrimidine, you have the same thing. You've seen Watson sugar, except that the part here, you see, if you take that as a reference, this part is different. So you have this wobbling. Glycosidic bond interaction. This is the normal cis base and the trans. Look at the position between the cis and the trans. You have the phosphate here. Here is up, here is down. Okay? That's very important. That is the normal one. When you have a trans, you can be sure that you cannot form a helical stem, but you can observe these in 3D structure to associate base to form 3-dimensional organization. In terms of classification and to grab up information from databases, it's very important because that you look for that type of conformation, and just when you pick up that, you are in a specific environment, and you can look what is happening. So that's another way. And then if you do that in a more simple way, you just put triangles, and that gives you 12 basic families. This is the reference that you will have where you can find the classification. And I will stop here, and I thanking also Eric for his contribution, Eric Westhoff, for his teaching in the same university as me, and then I used some of his background information for this presentation. I give him the credit for that because he's teaching the course in Strasbourg. And thank you.